So before we delve further into the talk, these are my disclosures relevant to this session. I'll give a quick overview of what tissue engineering is and how historically it has been used in different capacities. So as early as Neolithic period, sutures and other materials were being used as wound healing aids, and ancient Egyptians used linen and Europeans used catgut as suture materials. In ancient India, as early as 2500 BC, there are detailed descriptions of plastic surgeries where free gluteal fat and skin grafts were used to heal and to repair mutilations of ear, nose, and lips post-wars. Sir Isaac Newton proposed the idea that our body is a physiochemical machine and is the breakdown of that machine that causes the disease to happen. In the 18th century, Abraham Tremblay conducted some experiments with Hydra, if you remember from our high school or junior high school biology. When we cut one part of Hydra, it grows into an entirely new organism and so forth. So he was mesmerized with this regenerative capacity that these organisms have. Christian Pander in the mid-1800s proposed that cells proliferate and the cell environment and their interaction with the cell proliferation is vital for tissue regeneration. So this concept of tissue engineering is not new, but it's not concise. So there are many consensus papers and definitions that have laid down the stonework for what tissue engineering is and what needs to be done. So roughly speaking, there is a triad of for tissue engineering wherein there is biomaterials, cells, and there are growth factors. And one, two, or all three of the axes in combination can be used for successful and effective tissue engineering and regenerative medicine applications. So in this talk, I will try to showcase and uh, excite you with the story that happened and that started with a biomaterial and how it evolved into that becoming a growth factor with the core idea that we are stimulating these cells to get an optimal implantable engineered tissue. So before we delve further, as I mentioned in my talk's title, that I primarily look at the tissue engineering of musculoskeletal tissue. So there are specific challenges when it comes to musculoskeletal tissue because these are not homogeneous. These are highly varied tissues, and we have very hard structures in there, such as bone, and we have very soft structures, such as muscle, and we have fibrous tissues, such as tendons, cartilage, and intervertebral disc. And there is heterogeneity in their content, their function, and their organization, which compounds the problem because we have to personalize it and we have to restore the function along with the structure. If we don't restore or partially restore the structure, they are bound to fail again. There are many acute MSK injuries, and the tissue engineering challenges posed in many of these tissues, such as cartilage and intervertebral disc, is the limited blood supply, which makes it difficult to deliver these cargoes of growth factors and cell-based therapies. As we can see, the cell density increases as we move from softer to harder tissues, and whereas the vascularization also increases, and bone being the most vascularized tissue along with the muscle in the MSK system, whereas tendon, ligaments, and cartilage and intervertebral discs are in the state of relative hypoxia and relative hypoglycemia. Also, to add to the complexity, the mechanism of tissue injuries are also highly variable, and they range from chronic age-related degeneration to acute injury-related degenerations. So there are different elements in the musculoskeletal system that can be targeted for tissue engineering applications. And in my research experience thus far, I have targeted these four tissues. And primarily, there are two axes in my lab that we work on in term applications. One is for accelerated bone fracture healing, and second is in collaboration with Dr. Rosenweit about focusing on the tissue engineering of ligament and tendon tissue. There are numerous projects going in the lab, and as Tariq opened the talk, and for today's talk, we'll be only be talking about inorganic polyphosphates and how we are modulating them currently for fracture healing application. But I will try to give an overview that how the whole story came into being and how this is a very versatile molecule. So, inorganic polyphosphates, before we delve into that, it's very important to talk about calcium. And as I will show in my subsequent slides, how they're interconnected and how they superbly complement and manage each other. Some fun facts about calcium ion in the body. Calcium is the most abundant mineral in human body, which weighs approximately 1 kilogram in fully grown adult. And the 99% of the calcium found in the body is in calcium phosphate complexes. And non-bone calcium represents 1% of the total body calcium. And the serum calcium levels are very tightly regulated. And if you look at the range here, this is a very small range that the body works in. And if you go in the hypo or the hyper phase, there are very adverse reactions that happen in the body, including tetani and seizures. So this ion is one of the most regulated or tightly regulated ions in the body, which I will emphasize in forthcoming slide. Why is that? The non-ionized calcium is also bound to various proteins and ions in extra and intracellular pools to maintain this homeostasis in the serum and the bioavailability of calcium ions. And few of the commonly known proteins are also known to have calcium binding capacity, such as albumin, globulin, calmodulin, to maintain this homeostasis. So, what is the importance of calcium in our body? Almost everything we do is controlled by calcium, not in toto, but the major part of it. How our brain processes information, how we store memories. It's also needed for the heart to beat. Calcium is also involved in the active myosin cogwheel machinery to couple and decouple those molecules for movement. It's also used in neural transduction and neuronal firing. It also is used for neuromuscular complexes wherein the nerve sends the signal to the muscle to move via acetylcholine. It also plays a central role in the coagulation of blood when we have an injury. And of course, in bone and teeth formation and their subsequent homeostatic maintenance. There are numerous calcium signaling pathways and they are very complex and interrelated. And I want to draw your focus around this central portion, which is PKC, PKA mediated pathways. And we'll talk more about them in the coming slides. So this is very crowded slide, but I will try to make it as simple as possible. And this is the core concepts around calcium. So this incredible versatility in intracellular calcium levels, which are so tightly regulated, are because of this calcium signaling toolkit, as what it is called. And this ion can act in the context of space, time, and amplitude. I'm not trying to emphasize that these ions do time travel, but they do fall in the same or similar categories. What do you mean by space? So these calcium levels are divided into intracellular and extracellular compartments, and they can come from anywhere. If they're coming from, they can come from outside via these voltage gated and stress dependent channels, or they can come for the signaling pathway from within the intracellular stores from the store operated channels. Time. There is a baseline level of calcium inside the cell, and for it to act, it has something called a transient, which is an increase in calcium levels transiently. It could be a single transient, such as for releasing cell to cell these vesicles or the cargo from inside the cell, or for cell to cell signaling, or these cells or these fluxes could be sustained for a longer period of time to have a more profound and long lasting effect. And a prime example is the calcium cadmodulin interaction. And this phenomenon is called the frequency modulation. Whereas there is an amplitude modulation wherein the small amounts of calcium fluxes are there, or the amount of calcium inside the cell changes, which is actually less reliable because of the detection problems that we currently have to measure these calcium ions intracellularly. But the cells can appreciate and act accordingly, even with the modest changes in concentration of calcium ions. Calcium ions are, and the reason that it is very tightly regulated is that calcium ion can actually send a death signal if they are dysregulated. They maintain very critical processes in the cell, and if they are in excess, they go and they try to have a loop between endoplasmic reticulum and through mitochondria to sequester this extra calcium. But when this loop fails, it can actually cause mitochondrial death by sending the mitochondrial death signal and causing cell apoptosis. It is a very complex process. There are different calcium ions, ion channels present on the plasma lemma. There are also calcium ion channels present on the mitochondrial membrane, which are called the PTP or permeation transition pores, which we will delve more in when we get to the relevant sections. So what are inorganic polyphosphates? We use them daily in our daily lives, like they are part of oral healthcare products. Polyphosphates are also used in the bakery industry as preservatives. They're also used by the city to treat the deluge or recycle water and send it back to us because it acts as an ion chelator because of this high ionicity. Inorganic polyphosphates are nothing but linear polymers of phosphate units linked together by polyanhydrate bonds, and they can range from 2 to thousands in size. And the nomenclature that is given is a polyphosphate. 22 meaning there are 22 phosphate units attached to each other in a linear manner. Same way, polyphosphate 45, 60, 130 denotes the number of phosphate units that are attached in that polymeric sequence. You will come across these terms very frequently in the forthcoming slides. That's why I wanted to emphasize on the nomenclature. Polyphosphates were considered as the fossil molecules because these were the first molecules that were found or formed on the ancient earth when it was just a hot soup of boiling water. And the abiotic forms of polyphosphates are only found around the volcanoes where the magma or the lava goes into the sea, and that high temperature and that high phosphate content from the lava coming out actually makes these polyphosphates. These are ubiquitously found in all life forms on this planet, starting from viruses to plant cells to bacteria to mammalian cells. But in mammalian system, these were for the longest period of time were thought to be fossil molecules, which are the remnants of evolution, like your appendix, which has no apparent use. But recently, the focus has been shifted, and research has shown that these are not remnant molecules. These are actually very bioactive and they control very important life processes. The effect of these polyphosphates are chain and highly regulated and chain length and concentration dependent. But the mechanism of action of polyphosphate in mammalian cells is largely unknown. So in my previous engagement at University of Toronto with Dr. Kendall, I developed this polyphosphate detection method. There were other polyphosphate detection methods too, but it's kind of becomes very tricky to pick them up because they are available in the human or the mammalian systems in very small quantities. So the beauty about this detection system was that DAPI, which majority of us are aware, is a DNA binding dye. And what happens is that if you look at the DNA backbone, which is rich in phosphates, it has a homology with polyphosphate, which is also rich in phosphate. And it is postulated that actually the DNA molecules were, or initial single-stranded RNA and DNA were made on the polyphosphate backbone. So what polyphosphate does is it can also bind to DAPI, but instead of fluorescing blue, there is a fluorescent shift that causes it to fluoresce green. This is a very critical thing that we have to remember for the forthcoming slides because this is very important. I will show how we visualize polyphosphates in the cells. So based on this detection method, we were able to show that polyphosphate is present in all the tissues of the body: brain, lung, liver, kidney, heart, elements of musculoskeletal tissues. So they must have a role somewhere. They are just not there by de novo, or as I mentioned, as a process of evolution. I published a book chapter on the tissue engineering effects of or uses of polyphosphate in 2022. And when I was going through the whole literature, these are the different therapeutic areas where inorganic polyphosphates have been used in different formulations. This is the versatility of it. As a bioceramic glass, as insoluble polyphosphate, as calcium polyphosphate nanoparticles, and as retinol calcium polyphosphate nanoparticle. There are different formulations of this compound, but largely they were used as biomaterial without actually understanding how they work. They were just used as scaffolds or providers of phosphates, largely. So how the story began was that with Dr. Kanda's group, they were using the calcium polyphosphate amorphous bone substitute material. And what they observed was in bone defects, this beautiful layer of cartilage used to develop around these granular biomaterials. So when I joined the lab, I started growing this cartilage tissue and intervertebral disc tissue on these scaffolds. And this is just the tissue grown on this substrate in 2 weeks. This is like almost very thick implantable tissue that comes. So what we thought was that maybe these cells are responding in such a way because they like the architecture of the material, the porosity, there is some mechanical cue goings on. But it didn't make much sense because there is something more to the story. And we knew that this calcium polyphosphate material degrades, is biodegradable. And what it releases on degradation is soluble form of inorganic polyphosphate. So we knew there's some story around there that is triggering these cells. So previously in that lab, I had shown that the insoluble forms of inorganic polyphosphates at different chain lengths, such as polyphosphate 22 and polyphosphate 45, 60, and 130, had a dose-related response in GAG accumulation, which is glycosaminoglycan accumulation, which is actually a tissue engineering goal for this kind of tissue. We want to have as much matrix made by this cell to restore the structure and the function. And we also showed that this stimulates the outer peripheral cells of the intervertebral disc, and it triggers their metabolism and provides them with a phosphate unit. And the other student in the lab also had shown that when supplemented with inorganic polyphosphates with different chain lengths and different concentrations, it also increased the accumulation of GAGs and collagen type 2 in cartilage cells. So all these musculoskeletal cells were kind of responding to this polyphosphate, but we were not sure what exactly is the mechanism. So we were perplexed that it has to be more than just mere architecture of those biomaterials. So this was our key question. What is driving this anabolic effect? So for that, the story began with the media. So when we were supplementing polyphosphate and these cells in HAMS F12, the cells were growing okay. The constructs were by far not that exciting or I'd say attention catching. But as soon as we started using DMEM media, the polyphosphate supplementation had a profound effect. So this was observed by Dr. Sampierre, who is University of Ottawa currently, as a faculty member. So when we looked into it, it turned out that the major difference between these two medias is the calcium concentration. And the calcium concentration the difference is 1.5 millimolar. So what we started doing was that instead of using DMEM, we started using F12 and we started supplementing externally with calcium. So we used different doses of calcium, and what we saw was that both calcium and polyphosphates were needed for these cells to accumulate this matrix. Polyphosphate alone didn't have much effect, as we can see with the control baseline. The maximal stimulation to accumulate extracellular matrix was observed at 1.5 millimolar calcium ion concentration, which is same as the difference between F12 HAMS and DMEM media, and 1 millimolar of polyphosphate chain length 45, which is 45 phosphate units. When we supplemented these cultures with EDTA, this effect of GAG accumulation was completely blocked. There was no effect on cell proliferation, meaning that this matrix molecule that are being made is not because there are more cells that proliferated, it's because these cells were stimulated, and the same or similar number of cells were able to enhance the metabolic machinery to accumulate, or first of all make, and then accumulate these matrix molecules. Then we started to decipher or decode this mechanism of action. So initially our hypothesis was that polyphosphate is negatively charged. It somehow modulates the membrane of these chondrocytes, and because of which the calcium comes in through some ion channels. So what we did was we used this specific calcium ion channel blockers, and we could see and we observed that gadolinium, which is a stretch-activated calcium channel blocker, and nifedipine, which is a voltage-gated calcium channel blocker, they both blocked the effect or the anabolic effect of calcium polyphosphate along with calcium. But this was partial obliteration, not a complete blockage. So we knew there is some more side to this story. that there is some calcium that is coming from somewhere. It's not entirely the calcium coming from extracellular compartments. There has to be some other sources that are being released. And again, there was no effect of cellular proliferation at these doses of blockers that we used. So we next proceeded on to test out if we were to block intracellular calcium, how will it respond? So we used an intracellular calcium ion sponge, BAPTA-AM, and when we preloaded the cells, we put calcium and polyphosphate in the media. There was a flux coming from outside, and there was also intracellular calcium that was already sequestered. So we could see that BAPTA could also obliterate the effect of polyphosphate and calcium, but not completely. It gave us the clue that both these compartments are contributing these calcium ions along with under the effect of polyphosphate to be able to have that precise concentration intracellularly, which triggers the calcium signaling and which triggers the opening of the data boxes and accumulation of these GAGs. But how is polyphosphate facilitating this calcium ion entry, which was still elusive to us, to be able to trigger this anabolic effect on cartilage cells? So these are few videos, and I will just describe in quick 5 to 10 seconds what these are. This is calcium, live cell calcium imaging, that how calcium goes inside. And if you remember from the slide of space, time, and amplitude, these are actually fluxes that when you have live cell imaging, you're seeing that how calcium is going inside the cells in live time. So this is the baseline activity. And when we put polyphosphate chain length 45, you can see how these cells glow up. And many of the cells grow multiple times, like fire and fire and fire. Whereas chain length 130 didn't show much effect. But it's interesting if you look at the end of this video on this side, it stays calm for the large majority, but towards the late part of this observation period, it starts to fall. This is a delayed effect with polyphosphate chain 130. So when we quantified this flux data, so let me explain what flux is. So we capture images at every 5-second interval for a period of 60 observation, which is 300 seconds. And then we stitch them into the video that I showed you. But that fluorescence data that we get, we can actually quantify that because the microscope, we can get those raw numbers out in an Excel format. And it's a massive data set, but it can be cleaned, filtered, and processed. So what is a sustained flux? As I showed that some of the cells, they don't go like blink like this. They go blink, 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 one after another, repeated firing. As you remember from the frequency amplitude that I showed in the calcium space-time amplitude slide. So we define 4 consecutive fluxes happening as a sustained flux. And here you can see that when we tried different chain lengths, polyphosphate 22 and 45 was the one that were able to cause multiple fluxes. And many of the cells actually also experienced sustained fluxes. And this is the representative pattern of how these cells behave with calcium and polyphosphate. You can see that these cells at, say, polyphosphate chain length 45 is one spike. It goes on the baseline. Second spike goes to baseline, then again. And then this is the area where you can see there is a sustained flux, where it maintains the intracellular calcium concentration. So this is the characteristic of these fluxes. And if you can see and observe that this polyphosphate 130, it by and far remained low for the most period of time, but towards the end of the observation period, it starts to fire. There is something going on with that particular chain length, which actually is not bioactively active when it comes to cartilage cells. We did the same experiments and used calcium channel blockers, and we were able to partially block these effects. And same thing with the intracellular calcium sponge, which is BAPTA, which is an ion chelator, that it also blocked these calcium fluxes, meaning that whatever fluxes are going in, these are not the baseline intracellular fluxes that happens like while and you are talking. So these fluxes are actually being mediated by polyphosphate, and these can actually be countered and characterized. So far, so good. There is polyphosphate, there is calcium, and polyphosphate is facilitating calcium entry inside the cell. I don't know. We didn't know at that time how. We knew that, okay, some channels are involved, but those channels are not completely involved because complete blockage of those channels is not completely blocking the polyphosphate-mediated calcium entry. But what exactly happens to the calcium after it enters the cell? Because as I mentioned, calcium is highly tightly regulated. If polyphosphate is facilitating this much flux inside the cell, it will actually cause the cell, might actually cause the cell to die, can send a death signal to mitochondria. So what we observed was that when calcium enters the cell, we initially thought that polyphosphate is making in cohorts with the PHB or polyhydroxybutyrate channels, or membrane depolarization is facilitating the entry of calcium inside. We try to patch clamp these chondrocytes because chondrocytes are known to have a channel zone. They have different channels like sodium channel, calcium channel, potassium channel, glucose mediated sodium channels, but we were not able to successfully patch clamp because these cells in culture are very flat. But what we observed was that when we treat the cells with calcium and polyphosphate both, the polyphosphate also enters the cell. It doesn't stay outside because we thought it stayed in the membrane somehow, facilitated the entry of calcium, and that was its role. But this polyphosphate followed calcium and came inside the cell, and it sequestered itself into these vesicles. And as I had mentioned, that polyphosphate, when it binds to DAPI, causes a fluorescence shift from blue to green. So that's why we are able to visualize this molecule intracellularly. And we can see that post-exposure, these vesicles become laden with polyphosphate. And they're in very close approximation to the calcium stores inside the cell. And this calcium store that we suspect is corresponding to the endoplasmic reticulum sites. Further on, we observed was that in addition to these stores, these polyphosphate also migrated to the mitochondrial sites. And because we know that calcium in excess can send mitochondria death signal, so maybe these polyphosphates remain around calcium through weak van der Waals forces, so that the calcium cannot exert its negative effect on the mitochondria and not fool mitochondria or send a missed signal to mitochondria to send the death signal. And in further investigation and on further higher magnification, we also saw this polyphosphate to enter the nuclear membrane and in the nuclear sites. That was quite intriguing that polyphosphate doesn't just stay outside, it migrates all the way up to the nucleus. To see that how it works, so we preloaded the cells with DAPI and we marked the ROI like this, and we gave polyphosphate and calcium administration. And and as early as 30 minutes, we could see that the intensity of the nucleus started to increase, meaning that polyphosphate is actively going and increasing and sequestering, increasing that the signal intensity, which is quite a feat to be able to detect these levels of polyphosphate in live cell imaging inside the cell. And as I mentioned, that this was very specific because the excitation emission pairing is very specific. So it was not coming from DNA binding, it was specifically from polyphosphate binding. And when we only had calcium, only had polyphosphate administration, this increase and translocation to the nucleus site didn't happen. So we did some gene expression analysis and some radioisotope studies with sulfate or S35 to see and to quantitate the newly formed glycosaminoglycans. And what we found was that on calcium and polyphosphate co-administration, there was an increasing gene expression of aggrecan, SOX9, and collagen type 2. Those molecules that are actually needed for restoration of cartilage and intervertebral disc or central part of intervertebral disc function and structure. It also increased the GAG production, new GAG production, as evident from incorporation of sulfate S35. And these effects were actually obliterated or blocked by specific PKC pathway inhibitor calphostin C and cal module inhibitor W7 hydrochloride. So this suggested that this anabolic effect could be through the PKC calmodulin kinase CREB SOX9 pathway. We did some more studies, which I'm not going to go into detail for the benefit of time, where we showed with proteomics and phosphoproteomic analysis that those signaling pathways that I showed before via gene expression analysis are actually the signaling molecule pathways that are coming up. So it is actually calmodulin CREB mediated upregulation of gene expression for these specific ECM molecules. And in summary, Calcium and polyphosphates are required in chain length and specific chain length and concentrations. They are to be administered together for to have a beneficial effect. The activation of PKC calmodulin Krebs oxen pathway leads to increased GAG production. And polyphosphates, in addition to facilitating calcium entry and facilitating calcium signaling pathway, also translocates and resequesters around mitochondrial and ER sites, and also in the nucleus sites where it causes mitochondrial gene expression regulation. So in future directions, we are developing, as Tariq mentioned and showed Ryan's work, a customizable soluble polyp drug delivery vehicle, which I'm not able to show the actual formulation because of IP protection efforts. And we are also trying to advance the use of polyphosphate for tissue engineering application of other MSK tissues such as bone healing, which Tariq showed. And we are also trying to further explore the regulation of subcellular machinery by polyp, including the cellular bioenergetics. So just a quick 3 slides that how this fracture story is fitting in. So immune cells have something called pattern recognition receptors, or PRRs. And these PRRs have been classified into two subcategories, PAMs and DAM, which are pattern associated molecular patterns and pathogen associated molecular patterns and damage associated molecular patterns. So what happens is, if you have an infection such as open fractures, your immune cells recognize those foreign entities like bacteria and other infective agents and mount a response. Same way, there are many DAMs, or there are some ECM molecules that are exposed during injury, to which immune cells also host a response or mount a response, identifying them as not of this body. So these molecules or these DAMs actually have the potential to trigger the immune cells. So this is the not so comprehensive list of many DAMs, and we can see there are some subcellular components that can also act as DAMs when exposed to injury and insult. And among them, there are ATPs and some mitochondrial related proteins and some RNA, DNAs, and so forth. And they have the specific receptors that are present on the immune cells that can trigger these immune responses. And DAMs, the function is that they facilitate the migration, proliferation, and angiogenesis, which ultimately leads to tissue repair. So at the fracture site, the first thing that happens is white clot, which is a platelet clot, because it wants to arrest the bleeding, on which the RBCs come and they form the red clot and they arrest the bleeding completely. And then the process of fracture repair is initiated. So when the platelet is forming the white clot, from a resting phase, it goes to an activated phase and it starts to give out all its cargo, cytokines, chemokines, to attract immune cells to initiate the process. And one of the cargo is actually inorganic polyphosphates that are released in massive amounts. So the idea or the hypothesis we have is that, is polyphosphate acting as a dam at the site of fracture? Because there is tons of calcium at the site of fracture, there are tons of polyphosphate that is being released at the site of fracture. So if I have high enough calcium, high enough polyphosphate, why is it not biologically active? So that's why what Tariq showed or showcased Ryan's thesis work was that we are trying to create that effective delivery mechanism wherein we can have a maintained local concentration at the desired chain length and dose, so that it can exert its biological effect. And as soon as we want that anabolic trigger or that immune tactic trigger to be gone, it is exhausted and the delivery vehicle either dissolves or it assembles and remains there and act as a bioscaffold. So in acknowledgements, the polyphosphate work was done during my postdoctoral time in Dr. Kendall's lab in University of Toronto. And I was fortunate enough to be funded by the Arthritis Society, NSERC, and the University of Toronto Musculoskeletal Center for Fellowship Supports. And this is my current group. And the work that I presented was primarily and mainly done by Ryan here. And Tariq is carrying forward aspects of those projects. And last and not the least, these are my funding sources. Without their generous support and the support of my collaborators, this work and this exciting work would not be possible. So thank you for your attention, and I'm happy to take any questions.